Hello and welcome to the Fullagonist Pharmacology lectures. In this chapter, we'll talk about autonomic pharmacology. We're going to start with the anatomy of the autonomic nervous system, then explain cholinergic and anticholinergic drugs. After that, we'll study adrenergic and antiadrenergic drugs, and finally, pharmacology of the eye and treatment of the glaucoma. The autonomic nervous system is divided into sympathetic and parasympathetic parts. Parasympathetic nervous system is for rest and digest. Their effect on the organs are constriction of the pupils, slowing down heart rate, constriction of the airways, stimulation of the stomach motility and digestion, inhibition of the glucose release, and finally, stimulation of the sexual arousal in both males and females. Sympathetic nervous system is for fight or flight response. Its effects are dilation of the pupils, increase in heart rate, relaxation and dilation of the airways, inhibition of the digestion, stimulation of the glucose release, secretion of the catecholamines, promotion of the ejaculation in males, and vaginal contractions in females during the sexual intercourse. Anatomically, sympathetic nervous system arises from the thoracolumbar part of the spinal cord. Sympathetic preganglionic fibers leave the CNS through the thoracic, lumbar, and according to the new information, sacral spinal nerve. The parasympathetic preganglionic fibers leave the CNS through the cranial nerves, especially 3rd, 7th, 9th, and 10th. Previously believed that cholinergic nerves that innervate the pelvic organs, rectum, bladder, and reproductive organs are part of the parasympathetic nervous system. However, recent evidence suggests that the cholinergic preganglionic sacral fibers are actually derived from the sympathetic precursor cells, and when describing parasympathetic nervous system, instead of using old craniosacral term, we can use term cranial autonomic nervous system. Please remember the terms sympathetic and parasympathetic are anatomic. It do not depend on the type of the neurotransmitter release from the nerve endings, nor on the kind of the effect, which is either excitatory or inhibitory. Neuronal fibers that leave CNS form ganglia in periphery. Neurons before the ganglia called preganglionic neurons. Neurons after the ganglia are called postganglionic. All preganglionic fibers release acetylcholine. We call them cholinergic neurons. Acetylcholine is major neurotransmitter for both preganglionic and postganglionic parasympathetic neurons. Most postganglionic sympathetic fibers release norepinephrine, but some sympathetic postganglionic fibers release acetylcholine as an exception. We call that cholinergic sympathetic fibers. For example, sweet gland has such generation. Adrenal medulla is embryologically similar to the postganglionic sympathetic neurons. It releases a mixture of the norepinephrine and epinephrine into the systemic circulation. Final branch that I'm going to talk about is somatic innervation. Of course, it's mediated by the voluntary nerves. For example, innervation of the skeletal muscle and our voluntary movement. This transmission is happening by cholinergic neuromediator, acetylcholine. We need to be familiar with the receptors as well. Inside ganglia, there are nicotinic N-type receptors, and nicotinic M-type receptors are in skeletal muscles. Heart and smooth muscles are innervated by the muscarinic receptors of the cholinergic system, and alpha and beta adrenergic receptors by the sympathetic nervous system. And sweat glands, as an exception, are sympathetic but innervated by the cholinergic fibers and has M3 receptors. We'll clarify these in detail throughout this chapter. A final division of the autonomic nervous system is enteric nervous system. It's capable of acting independently of the sympathetic and parasympathetic nervous system. It's found in the wall of the GI tract from the esophagus to the distal colon and is involved in the motility and secretion. It includes the enteric plexus, also known as Auerbach plexus, and submucosal, also known as Meissner's plexus. The parasympathetic synapses may modulate enteric nervous system. If you cut parasympathetic innervation, you'll not diminish the activity of the enteric nervous system. The enteric nervous system contains large amount of the neurons that do not show histochemical characteristics for adrenergic or cholinergic neurons. They are called non-adrenergic, non-cholinergic neurons. They can be divided as nitronergic and peptidergic. The main mediator for the nitronergic neurons is nitric oxide, and in peptidergic we have peptides like substance P, vasointestinal peptide, calcitonin gene-related peptide, ATP, dynorphin, enkephalin, and serotonin, abbreviated by 5-hydroxytryptamine. There are two types of the cholinergic receptors, nicotinic and muscarinic. Nicotinic acetylcholine receptors are ligand-gated sodium-potassium channels. They are subdivided into the nicotinic N and nicotinic M-type receptors. Nicotinic N-type receptors are found in brain, autonomic ganglia, and adrenal medulla. Remember, adrenal medulla is derived from the neuronal crest cells. Embryologically and structurally, it's related to the neuronal tissue. That's why it has nicotinic N-type receptors. In the CNS, we also have same receptors. Nicotinic M-type receptor is found in the neuromuscular junction of the skeletal muscle. They play a key role in the neuromuscular signal transduction. You can easily remember these by mnemonic: N for neuronal cells and M for muscle. In contrast to the nicotinic receptors, muscarinic receptors are G protein coupled receptors, coupled with either GQ or GI protein. They are divided into the 5 types: M1, 2, 3, 4, and 5. To easily understand cholinergic receptors, we have a diagram which summarizes types of the receptors, localization, secondary messenger, and cellular and physiologic effects. M1 receptor is found in cerebral cortex, gastric and salivary glands. It's coupled with the GQ protein and leads to the formation of the IPA3 and D-acylglycerol. It serves higher cognitive functions and has a role in the emesis. M2 receptors are located in the cardiac tissue, primarily in pacemaker cells. It's coupled with the GI receptors. As you remember, GI is inhibitory and leads to the inhibition of the adenylyl cyclase and decrease in level of the cyclic AMP. Main physiologic effect is bradycardia, decrease in heart rate, and decrease in conduction velocity. One of the most important nerves is the vagus nerve. It innervates the heart and exerts its effect by the M2 receptor. It slows down the heart rate and can decrease the conduction velocity. That's why the parasympathetic stimulation slows down our heart rate. M3 receptors are present in the smooth muscle, like exocrine glands, eyes, endothelium of the blood vessels, lungs, sphincters, etc. It's GQ-coupled receptors, like M1, and activation leads to the parasympathetic effects like meiosis, accommodation to the near vision, bronchoconstriction, increase in the bronchial secretions, increase in the gastrointestinal peristalsis, relaxation of the urinary sphincters, salivation, lacrimation, and vasodilation. Acetylcholine can induce vasodilation by activation of the enzyme endothelial nitric oxide synthase and prostaglandin production. This effect is happening in healthy blood vessels and requires healthy endothelium. If the endothelium of the blood vessel is damaged, for example by the atherosclerosis, acetylcholine can induce opposite effect, a vasoconstriction. This is the pathophysiology in the vasospastic angina. We'll talk about that in details later on. M4 and M5 receptors are located in the hippocampus and substantia nigra. They have complex role and are not clearly defined. To summarize their cellular effect, M1, 3, and 5 receptors have similar secondary messenger system. They are GQ-coupled receptors. M2 and M4 are GI-coupled receptors. On this picture, we see a neuromuscular junction. On the right side, there is a light microscope picture of the neuromuscular junction. On the left side, we see electron microscope picture. We can easily appreciate the synapse itself, presynaptic membrane, synaptic cleft, postsynaptic membrane, and a lot of synaptic vesicles. This particular synapse is between neuronal and muscular cells, and such synapses are reason of our voluntary movement. Inside these tiny vesicles, there are main parasympathetic mediator, acetylcholine molecules. We need to know how acetylcholine is synthesized and how neuromuscular transduction is happening. Acetylcholine is synthesized by joining acetyl coenzyme A and choline. This reaction is mediated by the enzyme choline acetyltransferase. After that, acetylcholine is pumped inside the vesicles via the VAT transporter. The mediator is stored inside the vesicles, which avoids degradation and facilitates release. After the depolarization wave reaches the presynaptic membrane, N-type calcium channel opens and calcium enters inside the neuron. The amount of entered calcium is directly proportional to the released neuromediator. More calcium enters in the cell means more mediator is released. There are fusion proteins, vesicle-associated membrane proteins, let's call them VAMPs, and synaptosomal nerve-associated proteins, so-called SNAPs, or SNAPs. They serve to align vesicles with release sites, and they participate in triggering the release of the neurotransmitter. Released acetylcholine will reach to the postsynaptic membrane and activate its receptors. For example, in skeletal muscle, there are nicotinic M-type receptors here, which are ionic channel receptors. After binding, sodium enters inside the cell, and muscular cell depolarizes and contracts. By exactly this way, our voluntary muscles can move. In order to avoid constant stimulation and receptor desensitization, after binding to the receptor, acetylcholine is broken down by the enzyme acetylcholine esterase. Choline will be recycled back by the choline transporter and will be used later on for the synthesis of the acetylcholine. Several drugs and toxins work here. Hemicolinium blocks choline transporter and avoids choline recycling. Choline acetyltransferase is blocked by the bromoacetylcholine. Vesamicol blocks pumping the acetylcholine into vesicles by inhibiting VAD transporter. Amifampridine and 4-aminopyridine inhibit potassium channel efflux and prolong repolarization of the neuron. Amifampridine can be useful in the treatment of the Lambert-Eaton myasthenic syndrome. In this disorder, there are autoantibodies against calcium channels which lead to decrease in acetylcholine release and can cause muscle weakness. Amifampridine can inhibit potassium channel efflux and result in prolongation of the action potentials. This results in more calcium channel activation and increase in acetylcholine release. However, as a side effect, the drug can cause seizures, can prolong QT interval, and increase the risk of the arrhythmia as well. Botulinum toxin inhibits fusion proteins and prevents release of the acetylcholine from the presynaptic membrane, leading to the muscle paralysis. Botulinum toxin comes from the Clostridium botulinum. Its spores can be found in honey and alkaline environment, for example in canned foods. It can cause severe and life-threatening poisoning. But we can use botulinum toxin to treat blepharospasm, dystonia, strabismus, hyperhidrosis, and most commonly, it has wide cosmetic use against wrinkles. We have acetylcholine receptor antagonists. The inhibitors of the nicotinic acetylcholine receptors are called neuromuscular blockers and are widely used clinically, for example, in the anesthesiology and critical care. And there are inhibitors of the acetylcholine esterase, which inhibit degradation of the acetylcholine and increase their effect on the receptor. We'll talk about these drugs in detail in this particular chapter. Please remember that bromoacetylcholine, vesamicol, and hemicolinium have no clinical use. It means that you'll never see these drugs in the hospital. Neuromuscular transmission can be blocked by certain snake or spider-derived toxins as well. For example, tick paralysis is a toxin-mediated illness characterized by acute onset of the motor weakness. Several tick species can produce a toxin that inhibit neuromuscular transaction. For example, holocyclotoxins from Ixodes species inhibit voltage-gated calcium channels and inhibit release of the acetylcholine from the presynaptic membrane. Certain snake venom contains neurotoxins that affect neuromuscular signal transduction. These toxins are known as snake presynaptic phospholipase A2 neurotoxins. This group includes beta-bungarotoxin, notexin, typotoxin from the taipan snake, and crotoxin from the Brazilian rattlesnake. Toxicity leads to the massive release of the synaptic vesicles, and recovery requires nerve terminal regeneration, which may take weeks. Some snake-derived neurotoxin can also bind and irreversibly inhibit acetylcholine receptors on postsynaptic membrane. For example, alpha-bungarotoxin from Indian krat snake. Similar blockade is happening in case of the black widow spider bite. It has multi-component venom. One toxin is alpha-latrotoxin. It binds to the presynaptic membrane and leads to the depletion of the stores of the acetylcholine.